Good evening, all of you. So, the molecular biology chapter. Now we continuing the subchapter. So that is the transcription. So today chapter transcription is very important for the NEET and MSET level questions. So concentrated them. So first the transcription. So today discussing the chapter is what is the transcription. So the transcription means here clearly observe that point to point you understand here. So the process of copying genetic information from one strand of DNA into RNA is termed as transcription. So transcription means from the what is that? The genetic information is from one strand of the DNA into the RNA is termed as what is that? Transcription. So here only a segment of DNA and only one of the strand is copied. So because the reason is so very important. So why the only one segment, only a segment of DNA is so copied? So what is the reasons? The reason is, you remember here, if both strands act as a template, they would code for RNA molecules with a different sequence of proteins and amino acids. Okay, if two strands are act as a templates, they would code for the RNA molecules with the different sequences of protein and amino acids. This, and here you are observing one segment of the DNA would be coding for two different proteins, and these would complicate the genetic information transfer machinery. So next one is the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a failure one. So here 2 points are there. So here clear in a transcription means first you are observe that what is that? The copying genetic information from one strand of DNA into RNA. So this is the one key point, very important. And why the only a segment of DNA and only one of the strand is copied. So only one of the strand is copied. So that is the reason is, so what is that? The RNA molecules with a different sequence of proteins and the amino acids. That is the, and second one is the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a failure one. So these is the two reasons is the, these two reasons is explain about the what is that? The reason is only a segment of DNA and only one of the strand is copied. So that is the reason. These two reasons are very important for the level 2 question. And next here transcription unit. So in a transcription unit, in a DNA is consist three regions. So one is the promoter, second one is the structural gene, and the third one is the terminator. So dear students, you are observing the diagram. So number of time repeated questions, this is the transcription unit. So the questions is given on the basis of the diagram, and so clearly observe that here two strands are there. So 3 and the 5 and 5 and the 3 end. So here the what is that? So 3 and 2 or 5 and 2 3 end, 5 and 2 3 end is the stand is called as template stand, and the opposite anti-parallelly opposite stand is called as 5 and to 3 and is the what is that? Coding stand. So in this you are observing promoter is there, here structural genes are there, and terminator genes are there. So here the three, how many regions are consist the transcription unit? Three are there. One is the promoter, and the next one is the structural gene, and the terminator. The stands are two types. One is the template stand and another one is the coding stand. So this is the total. So consist the three regions in this. So immediately you are observing the, so how many regions are in the transcription unit means one is the promoter, and next one is the structural gene, and next one is the terminator. Okay, now here you are. 3 and 2 5 and polarity stand is called as template stand. Okay, so next one, 5 and 2 3 polarity stand and the sequence same as RNA is, that is called as coding stand. So here another you are observing the transcription and translation. Here you are observing this diagram is very important. You are observing here, this is the DNA. From the DNA, these are the coding stand. So first one, adenine, thymine, guanine, adenine, thymine, cytine. So this is the coding stand, and the opposite directions, the RNA is formed. From double stranded DNA to we can get the single stranded RNA. That time you are observing the stand is the coding sequence is changed. So here adenine, adenine common, thymine places here is what is that uracil is there. So from the two strand of the DNA, we can get the only single stranded RNA. So that's time you are observing. For example, this is the template stand, this is the coding stand. So in this template stand, so here what is the thymine? So here adenine, here adenine is there. So here uracil is there. Means AUG. This is the you are observing adenine, adenine same. Thymine places is uracil. Guanine places guanine. Adenine, adenine. Thymine places, only thymine places are changed. Instead of thymine places, here the present the what is the uracil. So these are the help for the recognition of the polypeptide chains. So this is the transcription. So here very important is the RNA polymerase enzyme is associates transitionally with initiation factor and the termination factor. So in the transcription, main important key role is the you are observing. What is that? In the 3 points are there. So coding stands 3, 500 to 3 and 500 to these are the diagram. Promoter, structural genes and terminator genes. And the next one, observe the what is that RNA coding is changed. So in a thymine places, we have to the what is that uracil is present. Thymine places is present the uracil, and remaining is what is that similar. So adenine, adenine places are there. So that transcript is the what is that RNA. So from the DNA to uh, mRNA, single stranded mRNA formation is called as transcription. So let us now here discussion about the transcription unit and the genes. So what is the meaning of the gene? First you are observing. The gene means what is that? The functional unit of inheritance, that is the DNA sequences. That is called as gene. So the gene means what is that? The functional unit of the inheritance. And next point here, the sequence coding for tRNA or RNA molecules also define a gene. Is a called as a what is that gene? So gene means what is that? This definition is also correct. These also correct. The functional unit of inheritance also called as gene, and the sequence coding for tRNA or RNA molecule also defined as gene. So here cistron. So that is the important cistron. Cistron means it is a very important cistron. This point is a segment of DNA coding for polypeptide. The segment of DNA coding for polypeptide is called as cistron. The structural genes in transcription unit can be said to be so monocistronic and the polycistronic or polycistron. So here first one you are observing monocistron. The monocistronic system is present in the eukaryotes. So that is the important interrupted coding sequence. These are the interrupted. These have to the what is that? The structural genes have the interrupted coding sequence. And next one, what is that? Polycistron. Polycistron is present in prokaryotes. It is the very important. The structural genes have the or the genes are the split. So the genes are the what is that split. So very important. Cistron is very important. Cistron means what is that? A segment of DNA coding for polypeptide, and structural genes in transcription unit can be said to be two types. One is the monocistronic. These are present in the eukaryotic. So this point is very important. Monocistronic present is eukaryotes. So the structural gene have the interrupted coding sequences. Next one is what is that? Polycistron. Poly PP. You are immediate easy remember it. Polycystonic prokaryotes. So the structural genes are split. Next, exons. What is the meaning of exons? Exons means the coding sequences are expressed the sequence. Expressing sequences is called exons. Here remember it. The coding sequences, expressed sequences are called exons. These are appear in mature processed RNA. These are interrupted by introns. So this is the very important appear in mature and expressed sequence. Next one, what is that? Introns. Intervening, these are do not appear. So these are the express sequences, exons are, but introns are not, do not appear in the mature, so processed RNA. So difference you are observing: exons, introns. Exons means expressed, introns means do not appear. So this is the very important, these two terms. And here cystron means what? Cystron means what is that? DNA coding for polypeptide, monocystonic eukaryotic, and poly is the what is that? Prokaryotic. So this cystron and the mono poly, these 2 points are very very important. Monocystron eukaryotes, polycystron is prokaryotes. So next one, here you are observing exons means expressed sequence, easy remember. Exon, ex, expressed. Introns means do not appear. So inheritance of character is also affected by the promoter and regulator sequence of the structural genes. Sometimes the regulator sequences are loosely defined uh, as a regulatory gene, even though these sequence do not code for any RNA and protein. So that is the important. So first you remember that these two words, so exons and introns. So this is the very very important. And the next one, some boxes are also there. So these are the Pribnow box. So it is a sequence of TAAT of, this is the very important, TAAT. So these are the 6 nucleotides that is an essential part of a promoter in bacteria like Archaebacteria. Next, TATA box, it is also called as Goldberg Hogness box. It is very important. TTA box is also called as Goldberg and Hogness box. It is DNA sequences 5 and TA, TTA, triple A, 3 and found in the promoter region in archaea and eukaryotes. Upstream of the transcription, upstream means what is that? 5 and to 3 and. Uh, Initiation site, place of unwinding, initiation, its function is associated with RNA polymerase 2. So that is important, polymerase, RNA polymerase 2. And the CAAT box, or it is also known as GC box, it is determine the efficiency of the transcription. So these 3 boxes are very important in the transcription. So TTA box is very important. It is also called as Goldberg Hogness box. Next one, types of RNA. So already the 3 types of RNA is present. mRNA, this is the provides template, and rRNA play a structural and catalytic role during the translation. tRNA read the genetic code and brings amino acids. All these 3 are needed to synthesis a protein in a cell. So this is the how many types of RNAs are there: mRNA, rRNA, mRNA, messenger RNA, rRNA, ribosomal RNA, T transfer RNA. So mRNA provide template, rRNA plays structural and catalytic nature role during the translation. tRNA reads the genetic code and brings amino acids. All three are, these three are needed to synthesis a protein in a cell. So here clearly discuss about this. These also very very important. These points is you noted and remember it. So the messenger RNA is reported by the Jacob Monade 1961. This is total cell total RNA. The range is up to 5 to 10%, is highly unstable. These RNA single stranded, and it is the straight and unfolded, and it is synthesized from DNA by a process known as a transcription. This molecular weight is, so 5 lakh. Lifespan in a prokaryotes up to, this is the very important, this point is, so up to in a prokaryotes, so 2 minutes, and the eukaryotes 4 hours. So here 2 minutes in a prokaryotes. These are the very, very important points. You remember it. And the next one, it contain the initiation codon AUG, AUG at the 5 end, and the nonsense codons. So also present UAA or the occur. Next UAZ amber, and UGA opal or amber present at the 3 end. 5 end is consist of the what is that initiation code, and third end is consist of the what is that nonsense codons. These are the three. So what is that? So UAE, UAG, and UGA. So this is the very, very important. UAA, UAG, UGA. And the next one, ribosomal RNA, it constitutes about the 75 to 80 percentage. And the next molecular weight is 10 lakh. And the next associated with the ribosomes. And the next one act as a seat of for the synthesis of polypeptide chain on its large subunits during protein synthesis. And the next one tRNA. So tRNA is the smallest RNA, also known as transfer or translator RNA, soluble RNA also. tRNA is also known as what is the soluble RNA, and it constitute about 10 to 15 percentage, and molecular weight is, so weight is 25,000. So the most widely accepted model of tRNA is, so it is a very important, tRNA is clover leaf model. It is proposed by the Holy. So tRNA is very important here. The smallest RNA transfer translator RNA. So this is the clover leaf model and the proposed by Holy. And transcription. So in a transcription, it requires an enzyme is called as RNA polymerase. So these whole enzyme, enter whole enzyme, whole enzymes means enter total enzyme, association with initiation factor sigma factor, and the termination factor rho factors. So alter the specificity of enzyme to initiate or terminate a transcription. So here two types of transcriptions are there. One is the in prokaryotes transcription, and the another one is the eukaryotes. In prokaryotes, and the second one is the in eukaryotes. Here, process of transcription, you are the diagram observe. It consists of the three stages: initiation, elongation, termination. So initiation. So in an initiation process, you are observing three stages: initiation, elongation, and termination. In a prokaryote is the, how many stages are there? Three stages are there. One is the initiation, second one is the elongation, and third one is the termination. Here clearly you are of the initiation. So RNA polymerase bind to promoter and the initiates transcription is called as initiation. So here you are observing RNA polymerase. RNA polymerase bind the here. Okay. So these are called as what is that? Initiation. So initiation means the RNA polymerase bind to promoter and initiate transcription is called as initiation. And next one, what is that? Elongation. Based on the sequence of nucleotides in the template strand, the ribonucleoside triphosphates are polymerized following the rails of complementarity. This is called elongation. And that during transcription process, only a only a short stretch of DNA remains bound to RNA polymerase. Here, the enzyme also facilitates the opening of helix and continues elongation in 3 and to 5 end direction of the template. So here you are observing. So in this diagram you are observing first the RNA polymerase is attached to the promoter, that is called as initiation. After this you are observing. So here elongation, the helicase is started this point, and they can separated the two strands and formed the and unwinding the so places in the between the strands. So that is called as elongation. So next one, last one is the what is the termination. In termination we can get the only one strand is comes out. So that is called as what is that? RNA. So in RNA they consist of the again rho factor, and sigma factor is the what is that? Initiated, and rho factor is the so fast to the helicase action. So here you are observing first in a three stages, you remember it: initiation, initiation stages is attached. Elongation means the helicase is increasing between the two strands, and termination places you are observing. So last one, when the RNA polymerase reaches to the terminator region, the RNA falls off and polymerase gets separated. So when the separation started in the elongation, separation is completed, that process is called as termination. So in a prokaryote transcription, we can summarize some points. They are, first one is, the mRNA does not require any processing to become active. So this is the first one. From these we can understand the what is the from the this prokaryotic transcription. Second point is, the transcription and transition takes place in the same component. So there is the no differentiation, that means no separation of the cytosol and the nucleus. So this is the very very important. So first point is, what is that mRNA does not require any processing to the become active. And second point is the what is the transcription and transition takes place in the same components, means no separation of the cytosol and nucleus. And next one, third is, transcription and translation can be coupled in a bacteria, so being much before the mRNA is full transcribed. So these 3 points are very very important. We can learn from the these experiment. So here simply you understand these based on to the diagram. Process of transcription in bacteria, first you are observing the RNA polymerase consists of the two factors, sigma factor and rho factor. So here you are observing when the RNA polymerase bind the promoter place, so that is called as initiation. And the in a elongation, that's time the rho factor is more effective in these stages, and the helicase enzyme activity is more, and they can separate the cut the bonds between the two strands. So helicase is increasing the helic helical structure in these places. So that is called as elongation. In a termination position, these helical started from the these place to, and these reaches to the end point. Then after these can separated and RNA can form. So this is the termination. So in this we can learn the key points. So that key point is very very important. So one is the mRNA does not require any processing to become active. And next one, transcription and translation takes place in the same component. So the no separation of cytosol and the nucleus, these two very very important. And the transcription and translation can be coupled in the bacteria, so because of being much before the mRNA is fully transcribed. So next one, transcription in eukaryotes. Now completed the what is the transcription in prokaryote completed. Next, transcription in eukaryotes. Here you are observing what is that. So copying, tailing, splicing. So these are the diagram. So you remember these one. So in these the transcription in eukaryotes, there are 3 RNA polymerases in nucleus in the addition to the RNA polymerase. The RNA polymerase can shows the division of labor. That is a very very important. So RNA polymerase can shows the what is the division of labor found in organelles. So that is a very important division of labor. So they are, first one, RNA polymerases, polymerase 1. So it contains the 5.8s, 18s, 28s rRNA synthesis. And second one is the RNA polymerase 2. So heterogeneous nuclear RNA, hnRNA. So very third one is the RNA polymerase 3, the tRNA, and 5s rRNA, snRNA, small SN means small nuclear RNA. So these 3 types of RNA polymerases are there. So very very important. These are, first you remember it. RNA polymerase 1 is 5.8s and 18s and 28s rRNA synthesis. So very very important. By heart it, which RNAs are present in the RNA polymerase are synthesized. So second one is the RNA polymerase 2. It's synthesizes hnRNA. HN means heterogeneous nuclear RNA. Next, third one is RNA polymerase 3. So it's contain the or synthesize the tRNA, 5s rRNA, snRNA, small nuclear RNA. So this is the RNA polymerase. In this slide, you are the remember these 3 types of polymerases. and the RNA polymerase shows the division of labor. Now, here your freshly synthesized RNA strand is called as primary transcript. So, from the DNA to synthesized fresh RNA strand is called as primary transcript. Primary transcript produced by the polymerase 2. So, that is the very important. So, primary transcript produced by polymerase 2 is called heterogeneous nuclear RNA or hnRNA. The primary transcript is converted into the functional mRNA after post-transcriptional processing, which is involves the three steps. They are called as copying, tailing, and splicing. So clear you are observing in a eukaryotes RNA polymerase is three, polymerase 1, 2, 3, and the here the what is the transcription freshly synthesized RNA strand is called as primary transcript. Primary transcript produced by the polymerase 2 is called as heterogeneous nuclear RNA, hnRNA. So this is the point is very important. hnRNA means heterogeneous nuclear. The primary transcript is converted into functional mRNA after post transcriptional processing, which involves three steps. So that is the one is the copying, second one is the tailing, and the splicing. So here, first one copying. Copying means the copying at phi end occur rapidly after the start of transcription. So an unusual nucleotide methyl guanosine triphosphate, methyl guanosine triphosphate, GMPPP, GMPPP. So methyl guanosine triphosphate is added to the phi end of hnRNA, heterogeneous nuclear RNA. It is catalyzed by guanylate transferase. Next, copy cap is essential for formation of mRNA ribosome complex. Translation is not possible if cap is lacking. So if cap is not there, automatically translation is not possible if cap is lacking, because cap is identified by so 18S rRNA of ribosome unit. So this is the copying. So here you are observing this diagram, you observe here. So in the diagram you are observing cap. This is a cap. So what is that? Methyl guanosine triphosphate. Okay. It is attached at the where in a phi end. So this process is called as what is that? Copying. Okay. Now you are observing tailing. So in a tailing you are observing. The tailing is the addition of adenylate residues about to 200 to 300 at three end in a template independent manner on newly formed hnRNA with the help of poly A polymerase. So here this diagram you are observing in this also. Okay, clear in this polyadenylation, poly A. So added to the what is that? Three end places. So cap methyl guanosine triphosphate adding the phi end, that is called as capping. So tailing means what is that? It is the where it is the added it the polyadenylation or polyadenylene is added at the three end, so that is called polyatyl. This process is called as polyadenylation. So next one, last one is what is that? First one copying is completed, and next one is the tailing is completed. It is the three end to template attached to the three end, and next one splicing. So here splicing is very important. The splicing is the process of removal of introns and the joining of exons in a defined order. Ribonucleoproteins are SN, SNRPs. So here your snRNAs means, so it is called as, this is carried out with the help of SNRPs, snRNA plus proteins. So the combination of snRNA and protein is called as SNRPs. And the SNRPs bind on either side of introns forming a spliceosomes. Spliceosomes. So this is the very important. SNRPs bind on either side of introns, forming a spliceosome, which is removed using energy from the ATP. The remaining RNA, which contains the coding areas alone, is called as functional RNA or mRNA. So the fully processed hnRNA is now called as mRNA, and it is transported out of the nucleus. So this point is very, very important. Transported out of the nucleus for the translation. So these are the key points. So copying, tailing, splicing. So this diagram is very, very important. Here you are observing copying and the tailing and the splicing. So this is the very, very important. Adding the cap, okay, methyl guanosine triphosphate, polyadenylation in a tailing, splicing, introns. So this is the very, very important key points. Now you are observing this total today topic. We are discussed the total. You are observing some key points are very, very important. After this we can started the questions. So here very, very important is what is that in a transcription. So first one, so only one strand is copied, so reasons. And the next one, the promoter structural and terminator gene, and the this diagram is very, very important. And the codon, the what is that, so coding strand and opposite strand, the RNA sequences. Instead of thymine we have to the what is that, uracil. And the next one, gene and the cystone definition, it is the coding for the polypeptide. And monocystonic eukaryotes, polycystin prokaryotes. And the exons means expressed, introns means do not appear. Next one box are the Pribonov box and the TTA box. It is the TTA box is very important. It is also called as Goldberg Hagness box. And the RNA. So in RNA, three types. These three mRNA, rRNA, tRNA. All these three are needed to the synthesis of a protein in a cell. So in this, so very, very important is the key point in a messenger RNA. It contains the what is that, it is observed the first Jacob Monode and the involvement to the transcription. And the prokaryote 2 minutes and eukaryote 4 hours contain the initiation codon and the nonsense codon. Ribosomal RNA in this the associated with the ribosome and the protein synthesis. tRNA is smallest RNA, so that is the very important. The tRNA model is clover leaf model proposed by Holy. So transcription, sigma, rho factors. In a prokaryotes, you are observing initiation, elongation, termination process are there. So in these eukaryotes, so observe the what is that. So process are very, very important in this RNA polymerase. So three are there. So in this copying, tailing, splicing is there. So the diagram is very, very important. Now we goes to the questions. So now which of the following RNA polymerase catalyzes the formation of transfer RNA in eukaryotes? So you are observing which of the following RNA polymerase catalyzes the formation of transfer RNA in eukaryotes. So here your RNA polymerase 1, RNA polymerase 2, RNA polymerase 192, RNA polymerase 3. So RNA polymerase 3 is the main important. So tRNA formed from the RNA polymerase 3. So that is the very, very important. RNA polymerase 1, RNA polymerase 2, RNA polymerase 192. RNA polymerase 3 is special. Remaining is prokaryote. So RNA polymerase is special for the eukaryote. So answer is D. Next, in the a segment of DNA coding form a polypeptides are called. So very important. A segment of DNA coding form a polypeptide. Polypeptide are called cistrons or transposons, and the exons are the introns. So already we know what is that, a segment of DNA coding. A coding form a polypeptide is the cistron. So answer is A. So next one, third, transcription refers to the, so first one, transfer of genetic code or sequences of DNA into RNA, and the formation of DMA from the RNA. So DNA, sorry, formation of protein. Next, polymerization of RNA in cell-free system. So here the what is the transcription? Already we know the transfer of genetic information or genetic code or sequence of DNA into RNA. So that's why answer is what is that? A. Next, fourth one. A transcription unit in DNA consists of so one is the promoter and the structural gene. Next, terminator and the none of this. So all are these present in the diagram already we know. So answer is, so you are observing A and C, B and D, D and A, B, C. So in this, what is the transcription unit in DNA consists the, what is that? In this, so D, all are these A, B, C. You are observing A, promoter, structural, and terminator. These three are present. So that's why answer is all of these. So answer is A, B, C. And the next intron is coding sequence and the non-coding sequence and the character of prokaryotic genome and the character of eukaryotic genome. So answer is what is that? Intron means coding sequences. Coding sequences. Next one, in this you are observing. So column 1, column 2, helicase. So gyrase, primase, DNA polymerase 3. So here joining of nucleotides, opening of DNA, unwinding of DNA, RNA priming. So here you are observing. So clearly 1A, B, B, 1B, second A, third C, fourth D. So here you are observing helicase, already you know. So opening the, what is the DNA, gyrase, so already unwinding of the DNA, primase, already you know what is that? Primase is the RNA priming, DNA polymerases. So what is that? Joining the nucleotides. So simple is there. So helicase is the, what is that? Opening of DNA, helicase. Next, gyrase means what is that? Unwinding the DNA. Next, primase means RNA priming. DNA polymerase 3 is joining of nucleotides. Next, the difference between mRNA and tRNA is so here mRNA has more elaborated three-dimensional structure due to extensive base pairing, and the tRNA has more elaborated three-dimensional structure due to extensive pairing. And the tRNA is usually smaller than mRNA. mRNA bears anticodon, but tRNA has codons. So here options you are observing. So you are observing A, C, A, B, C, D, B, C, and A, B, C. So here you are observing C, B, and C answer. You are observing tRNA has more elaborated and the three-dimensional structure due to extensive pairing. Okay, these also correct. And tRNA is usually smaller than the mRNA. So both B and C are correct. Next, ninth one. In eukaryotes, there are at least three RNA polymerases. Next, hnRNA has both exons and introns. See, hnRNA is formed in both prokaryotes and eukaryotes, and any mistake in DNA replication may cause mutation. So here, all are correct, and the all are incorrect, and the except C all are correct, and the only D is correct. So here you are observing C. hnRNA is formed in both, what is the prokaryotes and the eukaryotes. In a transcription, we studied the, what is that? hnRNA is formed in both prokaryotes and eukaryotes. So answer is here the, except these are the remaining, or hnRNA is the different means in a prokaryotes. In a chapter you are in this diagram you are observing hnRNA, we cannot discuss about here. So here diagram you are observing. That's why you are prepare the diagram, main diagram. So here you are observing hnRNA. So in this we can observe, but in the elongation in this process hnRNA is not there. So that's why. So here answer is only these are the separated is you are observing here. Okay, except C. HNRN is formed in both prokaryotes and eukaryotes. Remaining all are correct. And eukaryotes, there are at least three RNA polymerases present, and the both exons and introns are correct. Any mistakes in DNA replication may cause mutations. Next, ninth, the following is the schematic structure of transcription unit having some important component indicated by the A, B, C, D, in which of four options the components are identical. So this is the diagram already you know. So 500 to 300, 500 to 300. So this is the transcription start site. It is the A, here B and C, D. So here you are observing termination promoter, here the template stands and coding stands, promoter, terminator, coding stand and the template stand. See, promoter, terminator, template stand and coding stand. And the next one, yeah, terminator, promoter, coding stands and template. So here you are a promoter. A is promoter, B. So you are observing terminator. B is the what is the terminator. Next one is the what is that? C is the template. So D is the what is the coding. So that's why answer is C. So template, C, template, and the what is the, it is the promoter, template and the coding. So that's why here answer is the promoter, terminator, template stand and coding stand. So answer is C. 10th, refer to the given diagram. What does it represent? So this diagram already we know. So it is the what is that, methyl guanosine triphosphate is there, copying, tailing, splicing. These three steps are present. We can study it in the eukaryotic transcription. So that's why it is the represent the what is the transcription in prokaryotes, transcription in eukaryotes, and translation in prokaryotes and translation in eukaryotes. Answer is what is the transcription in eukaryotes. Okay, next. If the sequence of bases in coding strands of DNA is A, T, T, C, G, T, G, then the sequence of bases in mRNA will be. So here you are observing only what is that, thymine places is changed. So thymine places is changed means thymine. So uracil. Remaining all are similar. Only only thymine places is added to the uracil. So that's why answer is D. Next one, in transcription in eukaryotes, heterogeneous nuclear RNA is transcribed by, so RNA polymerase 1 and the RNA polymerase 2 and the RNA polymerase 3 and the all of these. So here hnRNA is transcribed by, so what is that, RNA polymerase 2. So this is the answer. So RNA polymer, heterogeneous nuclear RNA hnRNA is transcribed by the RNA polymerase 2. So next one, here the fully processed hnRNA is called as here one. Which one? So next, and is transported out of the second one, what is dash, into the third dash for translation. Here you are observing mRNA. So here you are mRNA, nucleus and cytoplasm. mRNA, cytoplasm, nucleus. tRNA, cytoplasm, nucleus. tRNA, nucleus, cytoplasm. Here you are observing. So mRNA, first one there. This is the answer. You are observing. The fully processed hnRNA is called as what is that, mRNA. Next one, what is that, and is transported out of the what is the, nucleus. So here nucleus into the which one, cytoplasm. What is the cytoplasm? So that's why answer is what is that, mRNA and nucleus and cytoplasm. And next one, last one, what is a split gene? What is that? What is a split gene? So split gene means here you are observing a gene with a croc in it. And next one, B, a gene which has no promoter. And the C, a jumping gene. A is the jumping gene, and the D is the a gene whose entire sequence are not retained in the mature RNA. So these are called as what is that? So split gene. So very important, a gene whose entire sequences are not retained in the mature RNA. So this is the very important. So in this transcription part, you are observing the, you prepare the main concentrate on the so RNA polymerase enzyme, RNA polymerase 1 2 3, and the next one these type. So what is that? The coding stand is the DNA uh, is there. So that stem. So he asking the what is that mRNA will be. So if asked the DNA, that time adenine place opposite stand is thymine. Thymine is there adenine. Thymine place is adenine. Here cytosine is there guanine. Guanine is there cytosine. Thymine is there adenine. Guanine is cytosine. So this is the when he asked the DNA, that time this is the correct answer. But here asking the what is that mRNA. mRNA means that time only thymine places are changed. Uracil. So these type questions and the diagram. So clearly observe the diagram. So the eukaryotic diagram and the prokaryotic diagram is, so sometime he asked the, so here given to the A, here given to the B. So A is represent the which one means copying. For example, here B given these places, it is represented the tail. So for example, these ring-like structure represent the C represented. What is that? Means splicing. So these type you are prepared the diagram. So and the next here also clearly these two types questions, and the another one is the these ones. So these are the diagrammatic questions are very important. You concentrate these sub unit and subtopic. So diagrammatic, mainly diagrammatic. So all of you, so prepare these, concentrate the what is the diagrams and the stands sequences, and the next one RNA polymerases. So this is the very important questions. All of you, thank you, thank you.